Bismillahirrahmanirrahim. Let's start the A-level biology 9700. Now this is the November 2020 paper 4-1. We have done in a previous video, we've done question 1, 2, 3 and 4. And in this one we are going to now do 5, 6, 7 and hopefully 8. Now let's look at this question number 5. This is a very interesting and a very difficult question, and so I want to take a lot of time in discussing this. Figure 5.1 shows a cat displaying the recessive phenotype. Now, this is something which you would have read, but you would have forgotten to really remember it. So, this is a cat which displays the recessive phenotype. So, whatever we are looking at, a picture might be a colored picture. I'm not sure what it was like in the exact paper. It is displaying the recessive phenotype for two unlinked genes. So this is again information they've given you to. So this cat is showing all the recessive traits. Now then, for more information is given. Number one, the first information given is the two information given. Big H and small h controls hair length. The allele for short hair is dominant. So short hair is dominant. So if it is dominant, as I told you before, it will be either this homozygous dominant or it will be heterozygous. Heterozygous is not dominant or recessive, it's just heterozygous. So HH controls the allele for short hair is dominant to the allele for long hair. So the long hair ones are all going to be small h, small h. Now this is something which you need to write on the paper and do this working. What is the dominant trait? What is the recessive trait? So this is definite, this is the recessive trait. Then big R and small r control coat pattern. And one is full color. This is the full color, and the full color with pigments on all parts of the body is dominant. So this is the dominant. So these are the ones which I've written on this side. These are all the dominant traits. Short hair number one, and full color, which is pigment everywhere. So the allele for full color with pigment on all parts of the body is dominant to the allele for pointed. So this is now, so what we have to understand is, so the allele for full color with pigment all over the body is dominant. So that will be what? Big R, or it will be big R, small r. So short hair is dominant and full color, which means pigment everywhere, is dominant, where the pigment is restricted. Now, where the pigment is restricted to face positive, dominant to the allele for pointed. Now here what do we have? So here we have the recessive trait is long hair, so that has to be small h, because it said here in the question HH. And then the pointed is the recessive allele. It's dominant to the allele for pointed. So that means pointed is recessive, and the recessive trait has to be small r, because that's what it said in the question. R, big R, small r. So pigment, and the pigment is on the ears and the face and the paws and the tail. So first let's get this clear. What is dominant? Dominant is short hair and full color. Dominant is short hair and full color. Recessive is long hair and pointed, which means there are, and then it said in the question that this cat is showing the recessive phenotype. This picture of this cat, figure 5.1, shows a cat displaying the recessive phenotype of the two unlinked genes. Part A of the question says, draw a genetic diagram to predict the offspring genotype and phenotypes when the cat in figure 5.1. So this cat, which is figure 5.1, which is what? Which is what? Which is this. So HH and this RR, because it says recessive. So HH and RR, this is this one here. So HH, long hair and pointed, is small h, and RR is crossed with a cat that is heterozygous for the hair length. So heterozygous for the hair length and heterozygous for the coat pattern. So heterozygous for the hair length would be big H, small h, and heterozygous for the coat pattern will be big R, small r. Now these letters are given in the question, so please use those questions. Do not add any letters from your own side. So this is big H, small h, and big R, small r, and here we've got that here. Look, you can see here this and this. So Long hair pointed crossed with a short hair full color, and then now you can see what will be the gametes be like. So this is the line for gametes. So then you've got the line for gametes, then we've got parent phenotype, then parent genotype, and then gametes. So you just write them separately, and you have to circle them. I just need to correct this. There's a mistake in this. So the gametes will be small h, small r. So you see, it's always 1, 3, and 1, 4, and 2, 3, and 2, 4. Now that is only one possibility, and that is small h, and small r. So this is what we have to understand, is that this would be the gamete. This would be the gamete. Now in this situation in which we have heterozygous for both, we have big H, big R, big H, small h, big R, and small h, small r. So we have four possible gametes. And then of course when we cross it, this is the ratio is 1 ratio 1 ratio 1 ratio 1. This is a famous one in which we say heterozygous for both crossed with the homozygous recessive for both will result in a ratio of 1 ratio 1 ratio 1. So you have to get this right, which is F1 genotypes, and then you have to get the phenotypes, right? It's because short hair, full color, short hair pointed, long hair, full color, and long hair pointed. So you had to use the words of the question, in which, as I previously explained it to you, is that you had to know what were the different dominant and the recessive trait, which I did this whole exercise on the top as I started this question. Now, coming to the B part of the question, this is again a very, very complex question, and you can see how it's a whole lot of written matter, is quite long. We need to understand this very carefully. I'll read it and then I will explain it to you, and I want you to do the same thing when you're taking the exam, inshallah. So, scientists isolated two sections of DNA thought to be correspond with the allele for full color and the allele for pointed R. So big R, small r. Sequencing these two DNA sections showed the DNA sequence for full color big R had a restriction site for the restriction enzyme HPA11. HPA, I'll just call it HPA. This restriction site did not occur in the DNA sequence small r because of a single nucleotide substitution. Information given to you in the question: DNA sequence, big R, small r, there's a restriction site for the restriction enzyme HPA11. The restriction site does not occur in the DNA when there is small r. The scientists then carried out an analysis of 3 generations of cats. Each cat was assessed for 3 features. Number 1, coat pattern, full color or pointed, the presence or absence of the HPA11 gene, the pair of alleles present at a variable marker locus thought to lie close to the RR locus. And the marker locus gene has 7 different alleles designated as 1, 2, 3, 4, 5, 6, 7. So there are 7 possible alleles. Like I told you before, blood group has 3 possible alleles. Blood group can be IA, IB, and IO. So there are 3 alleles for blood group. But of course, the combinations are more than, there are about 1, 2, 3, 4, 5, 6 different combinations, but 4 blood groups. So now we're looking at 3 things. Number 1, coat pattern. And you know, coat pattern is either full color or pointed. The presence or absence of the HPA11 restriction site. The pair of alleles present at visible marker locus thought to lie close to it, and the marker locus gene has 7 different alleles, 1 to 7. Now let's look at this chart which they have given you. Now, whenever you look at such a chart, there are a number of things that you need to see. Now, first thing that you need to see is you need to look at the key. Now, the key says full color male, squares are always males. Pointed male is the white box, and full color female is the black circle, and the pointed female is the white circle. Now, another thing which you've got to very important see is, now what is the dominant and what is the recessive trait? Now, how would I guess that? That is very important. If you cross two recessive, if you cross two recessive, all the offsprings are recessive. Now here, now this I've just given you an example. if you have small a, any letter you can take. I'm not saying take A. If you take small b, this is a recessive genotype, and you cross it with a recessive genotype, well, all the bachas, all the offsprings are going to be recessive. Now that is what you have to see here. When you cross this with this, all the offsprings are recessive. But when you have a dominant trait, like for instance, let's look at this trait, and it is heterozygous, then some offsprings will be having the dominant trait and some offsprings will have the recessive trait. Now see it in this diagram. Here now, let me just show you one of these examples. These are just examples which I want you to understand. So if I say this one is small a, and this is also small a, then all these are small a. But then let's look at another example. Now let's look at another example. Let's look at this example. Now I've shown you small a was this. Now this is big A, big A. Now all the offsprings will be this. But then how do we have this here? That means this is this. So you do this exercise on it, just like I've told you that if we have this crossed with this, all are this. Then we have this, if you look at this and this cross, this is also the same. So if any time you cross two recessive parents, all the offsprings will carry the recessive trait. So this is one thing that you must understand in genetics questions. You look at the pedigree charts, you look very carefully, you write some letters on it to explain to yourself, is this a recessive trait? And then you look up this chart, which is the full color, which is the pointed male, which is the full color circle, and the pointed female. And then you go back to the chart and you look at it again. Now let's look at the questions which are given to you on this pedigree chart. So let's look at the first question. It says identify evidence from figure 5.2 support these statements. The first statement was the pointed phenotype is due to a recessive allele. Two pointed parents always have pointed offsprings. Two full color can have pointed offspring. Well, if it's a dominant trait, full color, and then the pointed is the recessive trait, then two full color parents can have a pointed offspring. Then pointed offsprings have negative negative. So if you look at again this diagram, this is pointed, they have negative, this is pointed, they have negative, and the offsprings are also all negative. So this was the fact that you had to look at this part of the question. Then the part 2 of the question says big R small r gene is located on an autosome. That means it's not on a sex-linked disorder. It's not a sex-linked. Allele. So males have two alleles. Now in all these cases, which you say males have two alleles, and sons get one number from the father. And color of sons is not determined only by the mother, and gene is not on the X chromosome. Now, what does this mean? Males have two alleles. Now, if you look at all the males, now let's look at this male. Now, let's look at this male very carefully and look at it. Where did it get the 2 and the 4? Well, it got the 2 negative from here. This is the 2 negative it got from here, and the 4 negative it got from here. You remember that in males, then there's nothing on the Y chromosome. If it's a sex-linked disorder, the males only get the X chromosome, they get the Y from the father. So they should have only got one then from the father. So if you look at this diagram, this was a very nice one in which you could see that how this was. So let's go back to the mark scheme now. Males have two alleles. This one here, sons get one number from the father. Then color of the sons is not determined only by the mother, and gene is not on the X chromosome, because otherwise the father should have only had one allele. Then we come to the part 3. Part 3 says the marker locus and RR are closely linked. Now you have to see from the question, plus or minus and marker number inherited together as a pair. plus or minus and marker number inherited together as a pair. No crossing over seen. No plus 3, plus 4, minus 7 in the bottom right hand cat. Now let's look at the bottom right hand cat. Plus 3, plus 4 and minus 7. Now if you look at this bottom right hand cat, this is the one that we are talking about. There is no plus 3. So if this was going to be, we could have easily got this plus 3 if there had been crossing over. But there is no plus 3. Plus there is no plus 4. Plus 4 means what? Let's look at this now diagram. There is no plus 4. And there is no minus 7. Minus 7 meant what here? Now let's look at the minus 7. What are they trying to say? They're saying that if we had got some sort of a crossing over, then this could have come here, minus 7. Or here also minus 7. So there is no plus 3, there's no plus 4, there's no minus 7. Let's look at the mark scheme again. Plus or minus and marker numbers inherited together as a pair. No crossing over seen. No plus 3, plus 4, minus 7 in the bottom right hand cat. And this is what we were talking about, the bottom right hand cat. So there has been no crossing over here. And please now look at this again very, very carefully and understand what am I trying to say. Why did I say no plus 3, no plus 4 and no minus 7? So this was a fact which proved the point that the marker locus and R are closely linked. So they are both going together and there has been no crossing over. So they're both, if it is closely linked, then they both will go together. Closely linked means that both these are very near to each other. So if this is the gene for one and this is the gene for the other, so they will both go together and there will be no crossing over. They can't sort of cross over and then some genetic information is swapped between them. And these are called, that is why these are called the linked genes. That finishes this question. Next we go on to the next question. Question 6. Identify the precise location in a cholinergic synapse that is described in each of the statements. A region that contains many mitochondria, cytoplasm of the presynaptic knob. They're talking of the cholinergic synapse. A region where exocytosis of acetylcholine occurs, presynaptic membrane. A region that contains voltage-gated channel proteins, presynaptic membrane, and a region that contains ligand-gated channel protein, that is the postsynaptic membrane. Then outline the role of synapses. That was very easy. Unidirectional one-way transmission, communication between many neurons, interconnected neurons, and that's what's given in your book as well. Some insecticides contain compounds called organophosphates. Organophosphates can act as enzyme inhibitors in neuromuscular junctions. People who use organophosphates do not follow safety guidelines are at a risk of organophosphate poisoning, which affects muscle function. Suggest how organophosphates could affect muscle function in humans. Binds to acetylcholinesterase. They told you can act as enzyme inhibitors. So you know an enzyme we have talking about acetylcholinesterase. So binds to acetylcholinesterase, active site changes shape, acetylcholine not broken down, stays bound to the receptor, sodium channels stay open, sarcolemma stays depolarized, and the muscle stay contracted. So this is what would happen if acetylcholinesterase did not function and the acetylcholine did not break and go back into the presynaptic and be recycled. Again was a very direct question, very easy question. Outline the role of the sarcoplasmic reticulum in the contraction of striated muscle. Number 1, calcium channels open. Number 2, calcium ions diffuse into the sarcoplasm. You know, cytoplasm is called sarcoplasm. Sarcolemma is the cell membrane. So calcium binds to troponin, tropomyosin moves, cross bridges form, and myosin binds to actin. So any two out of these, and you got your 2 out of 2. 7. Structures and compounds involved in respiration and anaerobic conditions include pyruvate, reduced NAD, ethanol, carbon dioxide, cytoplasm, NAD, ethanol, lactate, oxygen, mitochondria. Complete table 7.1 by matching each description with one letter chosen from A to J to show the correct structure or compound. You may use each letter once, more than once, or not at all. So then as we go along, let's read this and I think of the answers. End product of glycolysis. End product of glycolysis was A, cellular location of respiration in anaerobic conditions. E, end product of respiration in anaerobic conditions in yeast cells was C or D. You could have written any one of them because you have to see there were 6 marks. So every line is 2, 3, 4, 5, 6. So end product of respiration anaerobic conditions was H. Gas released during alcohol fermentation was D. That was a very direct question, very easy question. Everybody should have got their full 6 out of 6. Then the B part of the question said respiration in anaerobic conditions in muscle cells during vigorous exercise can create an oxygen debt. Extra oxygen is breathed in after exercise to pay back the oxygen debt. Explain 2 ways in which the mammalian body cell uses this extra oxygen. Lactate converted or oxidized back to pyruvate or glucose or glycogen, and the hemoglobin is reoxygenated. So lactate, as you stop exercising, this lactate is taken to the liver where it is converted back to pyruvate, and the hemoglobin, which has lost all its oxygen, will be reoxygenated because by breathing in, like it says, each oxygen is breathed in, extra oxygen is breathed in after exercise to pay back the oxygen debt. To question number 8, and we read this very carefully. Palm oil is a vegetable oil that is used very widely in food products. The oil is extracted from the fruit of the oil palm tree. So oil palm trees have a higher oil yield than that of other oil producing plants. Figure 8.1 shows the oil yield of 4 crop plants: oil palms, olives, sunflowers, and soya bean. So here we have oil yield in kilograms per hectare per year, and here we have the crop plant. Read the table, read the graph very carefully, and then go on to the question what is asked on the graph. Calculate how many hectares of soya bean plants would be needed to produce the same yield of oil as 1 hectare of oil palm tree. Now you can see soya beans is 350 and the oil palm is 5,000. So 5,000 over 350, and it was an answer was simple. It was 14.3 hectares. Then another very direct question, which is a very easy question. Oil palm plantations in Malaysia and Indonesia have been created by cutting down rainforests. This reduces biodiversity. Naturally, you cut down the forest as a habitat for birds. Maybe some live in the trees, some live in the roots. There are so many organisms which live in the roots of the forest trees. Outline reasons why it is important to maintain biodiversity. Now you need to memorize these 5 or 6 points, and you can write these in any direct question. Aesthetic reasons. We love nature walks. We love going and watching forests and trees and rivers flowing and lakes and all that. We just love those. Then ethical reason. Why should we damage the earth? We should keep it for our future generations. We should maintain it. We should preserve it. Then there's a resource material, plants, trees, birds. They're resource material for a lot of things. Maybe they're medicinal value. Then ecotourism. Many countries rely on people visiting these places. And then, of course, the hotel industry works on that. The restaurant industry, the catering industry, the transport industry, which brings visitors to this place. Ecotourism. Then, of course, maintenance of food chains. If a certain tree dies, then the birds living on those trees or the caterpillars living on the leaves of those trees, everything will die. And it maintains the large gene pool. We have such a variety of trees and insects and plants, so gene pools are very important. And of course, forests are very important in mineral recycling as well. So this is what was expected of you in 5 marks. So 5 marks, I wanted 5 points. You should have written more so that maybe if one was wrongly written, you would get your mark for that as well. Then it said palm oil companies are now being asked to produce palm oil in a sustainable way. Sustainable way means that it can keep on and on for years and years and years. This means that no more deforestation should take place. Suggest ways in which individual consumers can encourage manufacturers to use palm oil from sustainable sources. So you educate people and you tell people do not buy unsustainable palm oil products. And of course, this would come out of a lot of awareness of the people. Educate for sustainable palm oil. So you advertise the media, social media, and you write and you say how we are damaging the environment and we should not buy this product or this product or buy this one more and buy this one less. And then, of course, if people donated to conservation charities and, of course, promoted that we should not damage the earth and we should keep all these palm oil from sustainable sources. So that was a very easy question, and that was for 9 marks. Now, as far as the essay questions goes, now this is the last year that this will be examined. This is now no longer in the syllabus anymore from 2022. You had to do only one question. Now, first of all, the first thing I tell you as you open the paper, you immediately go to section B. This is the rule for this year. You go to section B and you read both the questions and you now figure out which question are you going to do, because there's a choice. You either do this or you do this. Now, if you know one of these questions very well, then first you do this so that you get your 15 out of 15. But if you are not very sure on this question and you're sort of thinking about it, so just read the questions and then go back to section A and then come back here. But if you know one of these very well and you know say about 12 or 13 points out of the 15 marks, but then you better start doing this question. So if you know 9 better or 10 better, this is what you've got to figure out. So if you know 9 better, you start question 9 and you do this in the first 15 minutes of the exam. And if you do this in the first 15 minutes of the exam and you get say 14 out of 15, well, that's brilliant. That's brilliant. If you know this very well, you do this one, or if you know this one very well, you do this one. It's up for you to decide whether you're going to do question 9 or question 10. But if you are not very clear on this, then you go back to section A and you start doing this. And then while you're doing section A, maybe thoughts are going to come to your mind. And then when you come down to section B, you can come up with say 9 out of 15 if you are struggling with this. But of course, these are essay questions are a little easier, and I feel everybody should aim to get good marks in the essay questions. And then you do this either first or but you read this first. As soon as you open the paper, you go to section B and read the question and decide which ones are you going to do. Because even if you don't do it, it goes well because you keep on thinking about it at the back of your mind, and when you come back to it, you will actually do a better job at it. So that finishes this paper, and I hope this has been helpful. And best of luck with your exams.